Okay, so picture this. You're a biomedical researcher today, and you are just swimming, maybe even drowning in data. I mean, millions of research papers, clinical trials popping up constantly, patents. It's this huge flood, way too much for anyone to handle manually. And that means, you're probably missing vital connections, feeling swamped, maybe falling behind. So for this deep dive, we're asking, how can you get a shortcut? How do you stay genuinely informed in all this noise? Today, we're looking at one potential answer, the SWALife literature mining tool. SWALife Biotech, they have this really compelling mission focused on helping cancer patients using novel plant-derived molecules that target DNA damage response, pretty specific stuff.
Yeah, and what's key about a tool like this is not just about, saving time or being more efficient. It's really about taking all that raw text words into paper, basically, and transforming it into structured intelligence, something you could act on. Think of it like turning this giant, messy pile of puzzle pieces into a clear map. It fundamentally enables research in a way that just wasn't possible before for you.
Okay, that makes sense. So if computers are reading all this dense scientific stuff, how does that actually work? What's the tech behind it?
Well, the core of it is natural language processing, NLP. That's the field focused on teaching computers to understand human language.
Okay.
It involves cleaning the text first, breaking sentences and words down. Then comes the really clever part, named entity recognition, or NER. That's spotting the key things like specific genes, diseases, drug names.
Okay, like pulling out the nouns that matter.
Exactly. And then relationship extraction, RE, figures out how they're connected. Does drug A treat disease B? Or does gene C cause condition D? So you instantly get these connections mapped out, connections that might take, well, months to find reading manually.
Right.
But, and this is important, biomedical text is uniquely tricky. You've got these incredibly long, complex sentences, lots of ambiguity, different terms used for the same thing.
Oh, yeah, I can imagine.
So you can't just use some off-the-shelf AI. You need highly specialized models, custom trained on this kind of language to get reliable results.
Okay, that's crucial. So you extract all this info, the entities, the relationships. How does that become genuinely useful? It can't just be a massive spreadsheet, right?
Oh, exactly. That's where knowledge graphs come in. All those extracted bits get organized into these interconnected networks. Picture a huge smart web where every drug, gene, disease is a node, and the lines show how they relate based on the literature. It mirrors how researchers actually think about connections.
Like a concept map, but interactive and massive.
Precisely. And critically, these graphs are structured using ontologies. Think of them as standard dictionaries or vocabularies. They make sure that when one paper says heart attack and another says myocardial infarction, the system knows they mean the same thing. It ensures consistency, so you can compare apples to apples across all that data.
Got it. Standardizing the language. So how does AI push this beyond just mapping out what we already know? Where's the discovery part?
This is where it gets really exciting. AI and machine learning allow for what we call generative discovery and predictive optimization. By learning patterns from truly vast amounts of data, the AI can start to suggest new things.
New things, like what?
Like novel drug candidates or previously unknown links between a gene and a disease. Things a human researcher, even an expert, might totally miss just because of the sheer scale. It's not just finding existing facts anymore. It's helping you generate brand new hypotheses to test.
Wow.
This dramatically speeds up the early stages of drug discovery. You see tools like AlphaFold 3 predicting protein structures. That helps optimize how potential drug molecules might interact. It tackles that huge bottleneck of finding genuinely useful biological insights, basically giving you a head start.
That's really powerful. So how does SWALife apply this specifically to their niche, natural products from plants?
Right, SWALife's tool is likely tuned for that. It doesn't just use standard databases like PubMed or clinicaltrials.gov. It integrates specialized databases too, things like HRB 2.0, which focuses on herbal medicine and traditional knowledge.
Okay, so blending old and new knowledge sources.
Exactly. This allows it to grapple with the complexity of natural products. They often have multiple active compounds, hitting multiple targets in the body, affecting multiple pathways. The tool helps decode those synergistic effects, which are often missed when you only look for single magic bullets. It gives you a much more holistic picture.
Can you give a concrete example? How might this uncover a hidden relationship for someone using the tool?
Sure. There's a case study related to major depressive disorder, MDD, using network analysis on this kind of integrated data, herbs, compounds, gene targets, diseases. The tool did two things. First, it confirmed some known herbal remedies for MDD.
Okay, validation.
But second, it actually highlighted two potentially novel herbal compounds. and suggested specific biological pathways they might be working through, pathways that aren't previously linked. That's not just finding info, that's actively pointing towards new research directions. Years of potential discovery, time saved, giving you a real edge.
That's pretty amazing. But I mean, dealing with all that data, especially maybe traditional sources alongside clinical trials, it must be messy. What are the big challenges?
Oh, absolutely. Challenges are significant. Data quality is always an issue. Inconsistencies, errors, the ambiguity in language we talked about is huge. And just the sheer scale, processing billions of data points. You need really robust, scalable algorithms and sophisticated AI that can tell the difference between a real biological signal and just, a random correlation or a spurious hit in the data, making sure the insights you get are genuinely reliable and actionable. That's the constant focus.
Right, separating the signal from the noise.
Precisely. So, wrapping up, SWA-Life's tool and others like it, their main contribution is turning this overwhelming flood of information into structured, usable intelligence. For anyone listening who works in, say, natural product drug discovery for cancer, having this capability is a serious competitive advantage.
And looking ahead, where does this technology go next? What should we expect?
We'll definitely see deeper AI integrations, think more powerful generative models, maybe even AI suggesting entirely novel chemical structures for you to synthesize. Also, integration with other data types, multi-omics, combining literature insights with genomic data, proteomic data, giving you that complete biological picture.
A truly holistic view.
Yes. And imagine real-time analysis, getting insights almost instantly as new papers are published.
Constant updates.
Yeah. Which leads to a final thought for you, the listener, to maybe chew on. Think about how these tools are fundamentally blurring the lines. The line between just analyzing data and actually doing scientific experimentation. Research is shifting, moving from purely observing and testing in the lab towards a much more predictive approach, where AI generates strong hypotheses first. What used to take years of painstaking trial and error might soon, maybe, be revealed through just asking the right question, running a clever query on one of these systems. Food for thought, isn't it?